A little late today. I had an interview. I was interviewing students for something I had to be at, but I'm here. Everybody ready for a weekend? Yeah, let's get this over with, huh? Okay. I'll do what I can. So, want to finish talking up about nucleic acids in general, then we'll start turning to replication of those nucleic acids, okay? So, one of the first things to think about is something that I have mentioned earlier, and that is the size of DNA. The size of DNA relative to a cell is pretty remarkable. As I mentioned, our cells each have 7 feet of DNA in them, and that's a remarkable amount of DNA, and it tells you that something very special has to be done to compact them and make them fit into the environment that a cell is in. Okay? One of those one of the ways in which this is done is through a coiling process that makes a structure we call chromatin. Chromatin, okay? So chromatin is a sort of a structural part of a chromosome. Chromatin is a structural part of a chromosome. What does that mean? Okay? Well, when we say chromosome, one of the first things people think of is DNA, right? And chromosomes are much more than just DNA. That's important to understand, okay? Chromosomes are comprised of DNA plus some very basic proteins, that is positively charged proteins, called histones. Now, what I'm describing for you with respect to chromosomes and chromatin and histones is all true only in eukaryotic cells. Bacteria don't have chromosomes as such, at least not like we do, and they don't have histones, and therefore they don't have chromatin. So we can think of bacteria as having sort of naked DNA, whereas our DNA is so humongous that we've got to coil it up to fit inside of stuff. So that's what the histones provide. The histones are schematically shown on this figure as little round balls, okay? So we can think of like a ball of yarn that we're wrapping around there, and the DNA is the green stuff that's wrapping around there. Now, you'll notice I described the histones as having a positive charge, and that turns out to be really useful because the DNA in its backbone, you learned on Wednesday, has a negative charge. So they're attracted to each other, and they're held fairly tightly together. And not only is the DNA wrapped around the histones, but those wraps of DNA in the histones themselves are wrapped. So we see different levels of wrapping that occur. So we make some coils, and then we take those coils and we coil them with other coils. And it's through all of this coiling that's going on that allows the cell to fit all of that DNA into the confines of the nucleus. It's a really remarkable thing that happens. Now, one of the things that we'll talk about soon is the conversion of DNA to RNA, that is the process of transcription. We also think about the replication of DNA. And in both of those processes, we have enzymes that have to get access to the DNA, and they have to unwind those strands, and they have to either copy the DNA or they have to make RNA from them. And you can imagine that if I have coils of coils of coils of coils of coils of coils of coils, which is what I have with chromatin, that I'm going to have some challenges in identifying the sequences that I want to copy into RNA, how I can go through and copy the DNA. And in this course, we will only very briefly talk about those things, but I want you to think about that. The logistical issue of finding a sequence very deeply embedded within a chromosome and then copying it, or in some cases, I say, replicating the entire chromosome itself. It's a very, very big challenge. Okay? So this is an important thing for us to keep in mind. Histones occur in pairs of 4 pairs of 2. Okay? The ones that we see here are called, this is a what's called a core particle. You don't need to know that. But these histones within here, there are 8 proteins, 4 copies of 2 each. Okay, that includes what are called H2A and H2B. This is the names of the histones. H3 and H4. And you might wonder what happened to H1. And H1 is not part of that core, but H1 appears to play roles in between the individuals out there. So there are 5 histone proteins, 4 of which in pairs are in that core sphere that you see here. Okay? All right. Another thing that we think about with respect to DNA, and this is getting off the subject of chromosomes, but it's a related topic, and that is the strands themselves. The strands of DNA are held together by hydrogen bonds. I want you to remember that. 3 bonds each for GC base pairs and 2 bonds each, 2 hydrogen bonds each for AT base pairs. That's the only thing, or those are the only things that hold the strands together. Okay? Now, the more base pairs we have, that is, the longer the double-stranded helix of DNA is, the more of those AT and GC base pairs that we have, the more hydrogen bonds we have holding it together. But the point is that there's nothing more than hydrogen bonds that hold the strands together. Now, I tell you that because we can actually take the strands apart. And this really isn't anything different than what we talked about with respect to denaturing proteins. I said when we denature proteins, we're unfolding the proteins. And I said we could do that with heat, we could do that with pH, we could do that with detergents. There was a variety of ways that we could denature proteins. And there's 2 very common ways we can denature nucleic acids. And they rely on the fact that the forces that hold together the nucleic acids are hydrogen bonds. The 2 things that can affect those hydrogen bonds, not surprising, are the same 2 things that affect the hydrogen bonds in proteins. And they include heat and they include pH. pH is going to change charges. Heat is going to directly affect the hydrogen bonds. Now, it turns out that if we take DNA, it doesn't matter how big the DNA is, but if we boil that DNA, we can pull the strands apart. It doesn't matter how long it is, because each hydrogen bond is unable to outdo the heat of the boiling water. So what we see for a DNA molecule, and what you see on the screen here, is a plot of absorbance versus the temperature of the solution that the DNA is in. And you see something interesting happen. It turns out that DNA absorbs light pretty strongly at a wavelength of about 260 nanometers. 260 nanometers. If I start out with double-stranded DNA at about 70 degrees centigrade, and I start heating it and heating it and heating it, I all of a sudden start seeing that the light that's being absorbed by that DNA increases. That is, the DNA is absorbing more of the light. Okay? And what's happening during that increase is the strands are coming apart. It turns out single strands will absorb light way better than double strands will. The midpoint of that transition from double-stranded to single-stranded, which is what we have at the top, is called the transition temperature. It's also called the TM, and you may call it the melting point. They're all the same as far as I'm concerned. Okay? Now, that turns out that taking apart of the strands turns out to be a very important consideration for a process that we'll talk about later called the polymerase chain reaction, or PCR. So I'm only introducing it to you right now. I will talk about that technique and that process later in the course. Okay. That's just some handwritten notes. Okay. Well, the last thing I want to say about nucleic acids relates to RNA. And RNA, of course, is related to DNA. With RNA, we see that there are different types of RNA that's there. And there are 3 primary types that we'll be concerned with in this class. And they're the 3 that you typically are concerned with in a basic biology class. That is transfer RNA, ribosomal RNA, and messenger RNA. And these 3 types of RNA all play central roles in the synthesis of proteins. They all play central roles in the synthesis of proteins. The other ones that are listed on here are more recently discovered DNAs that, when they were discovered, were considered to be pretty trivial and have little or any role, but now we know they actually play some very big roles in controlling whether or not a given protein is made. Now, that's actually a topic for a more advanced class than what we have. I will mention them later in the course because they're actually kind of interesting for us, giving us ways to control cells and what cells make. Okay? But I'm not going to talk about them here. I'm going to focus on the first 3, which are transfer, ribosomal, and messenger RNA. Okay? Well, I'm sure you learned in basic biology that transfer RNA is a molecule that looks something like what people draw as a clover leaf. And that clover leaf has 2 main regions that will be of interest to us. Now, this is the complete structure of a transfer RNA. You can see that it has a 5' end, and it loops all the way around and comes back over here and ends at a 3' end. Meaning that what you're looking at is one strand of RNA. The base pairing that's happening is happening because the strand is forming pairs within itself. It's not forming pairs with another strand. Now, the 2 areas of interest to us are the 3' end up here at the very top, where it says 3' OH. And the other is this loop down here, which is called the anti-codon loop. That's this region labeled number 2 at the bottom. The anti-codon loop. Now, the reason we're interested in the 3' end is because it's at the 3' end that an amino acid becomes covalently attached. Transfer RNA has the function of transferring amino acids to the place where protein is being made. Transfer RNAs have the function of transferring amino acids to the place where proteins are being made. Yes? So the question is, what's in between loops? These are simply base pairs. So you could have, if you had on the left an A, on the right, then you would have the complement, which would be U. Right? So they're just like you would see in 2 different strands, but now they're all forming within one strand. Okay? Sure. So transfer RNAs have the function of transferring amino acids to the place where proteins are being made in the cell. That's why they're called transfer. Now, I said that at the 3' end, that's where an amino acid is attached. Which one? Well, there are 20 different amino acids used in making proteins. And it turns out that which one gets attached up here is determined by the sequence of what's down here in the anti-codon loop. This is literally the foundation of what we call the genetic code. So there's a link between which amino acid up here at the 3' end and what the sequence is down here. Now, we'll talk about that later. But these 2 things are aligned, meaning that the sequence determines what amino acid gets attached. Now, I'll talk about translation later, and as I say, I'll talk more about that then. Okay, the second type of RNA that I want to concern ourselves with is messenger RNA. Messenger RNA is called messenger RNA because it carries a message. That's what a messenger does. A messenger carries a message. The message is also related to the genetic code. Okay? We'll see how that relates when we talk about translation later. But the message, that is the messenger RNA, is made by copying a sequence within the DNA. So the DNA has a sequence in it that gets copied and made into the messenger RNA. The messenger RNA is carrying that sequence to the place where protein's being made. So we see the transfer RNA is carrying the amino acid. We see that the messenger RNA is carrying the message, that is the information necessary to make that protein. I'll repeat that. It's a very important point. The messenger RNA is carrying the message, and the message is the information necessary to make a protein. So now we've got two of the players necessary for making proteins, the amino acid and the information for how to put the amino acids together. The third component is of the RNAs that I've talked about is called the ribosomal RNA. Okay? Now, you saw the tRNA had this cute little clover leaf-like structure. It's fairly small. It's about 100 base pairs or so in length, 100 bases in length or so, all right? This is a ribosomal RNA, and you can see the letters are very tiny because there's on the order of about 1,500 nucleotides in this particular ribosomal RNA. I couldn't get the word out, the ribosomal RNA. And you can look at it and you can say, and this is also, by the way, just like the tRNA, this is one strand. You can see that the strand is pairing with itself. And if you think about it, this isn't something unlike what you've already seen, although you're seeing it in a different way. Yes? You're actually, this is good. He's actually anticipating what I'm getting ready to tell him. Okay. What's causing this structure? What causes the circular regions? What causes the base pairing? What causes all of these things to occur? The simple answer is that the bases are aligned so that they have a sequence that they're complementary to another portion of the same messenger RNA. Meaning GGGG lines up with CCCC. Right? So if I have one part of the messenger RNA that has a string of Gs, and I have another portion of the same RNA that has a string of Cs, if those two guys bump into each other, they're going to form base pairs. So it happens as a result of what we call self-complementarity. Self-complementarity. I'm going to tell you something that's probably going to be an extra credit question on the next exam. Okay? And that is the proper spelling of the word complementary. All right? When I say the word complementary here, it means that two things fit together. The two strands of DNA, for example, are complementary. The G goes with C, the A goes with T, et cetera, et cetera. In RNA, the A is going with U and the G is still going with C, et cetera. And they're complementary. But that's completely different from me complimenting you on the way that you're dressed or the way that you did great on an exam. Right? Complementary, that means when I'm complimenting you on something good quality, that's complementary. And If you spell that C-O-M-P-L-I-M-E-N-T-A-R-Y, that means that you are complimenting someone. If you use that word to describe what I've just described here, you will not get that extra credit. So I'm going to spell the complementary that is the word that you should use to describe how the strands come together. I want to see everybody get this right. If everybody gets this right, you will be beating seniors in biochemistry and professors in biochemistry who frequently get it wrong. Is that a good challenge? Okay, here is my challenge. The correct spelling of putting the two strands together, complementary, is C-O-M-P-L-E-M-E-N-T-A-R-Y. Everybody's laughing, but it's fine. This is a serious matter, okay? This is a serious matter. C-O-M-P-L-E-M-E-N-T-A-R-Y. The only difference between that one and the other one is this has an E after the L. The other one had an I after the L. All right. But I will tell you, the reason I bring this up is there's nothing more embarrassing than reading a very highly technical blah, blah, blah that somebody has written about nucleic acids, and they spell it with an I, and you're going, what a bonehead, right? Okay, so don't be a bonehead. That's important. All right. Now, so your question, and getting back to his question over here, how does this form? It happens because of complementarity. But if you think about it, I told you that a protein folds. And I told you that folding was a function of the sequence of amino acids, which meant that there were some hydrogen bonds that helped the secondary structures to form. There were folds that helped this to happen. And it got held into a tertiary structure, right? That tertiary structure was essential for the protein's function. This guy folds into a tertiary structure. Just like a polymer of nucleic acids can fold into a tertiary structure, so too can a polymer of ribonucleotides fold into a tertiary structure. And you start thinking when I tell you that this is as big or bigger than most proteins. Is it possible that the folding of this structure might have some catalytic function like the folded protein has? And the answer is, in some cases, yes, it does. It forms a catalytic molecule in some cases, not all cases, but in some cases it forms a catalytic molecule we call a ribozyme, R-I-B-O-Z-Y-M-E. A ribozyme, if I were to ask you on an exam what a ribozyme is, I would hope you would tell me it's a catalytic RNA. It's an RNA that catalyzes a reaction. There are several kinds of catalytic RNAs that we know. They're not real common, except for one I'll tell you about. They're not very common. Okay? But the one I'm going to tell you about is one of the most abundant catalysts on the face of the earth. It's the one that catalyzes the formation of peptide bonds. The formation of peptide bonds is catalyzed by a folded ribosomal RNA. And that happens within the confines of a bigger structure we call the ribosome. So we have ribosome, ribozyme, different things. Ribozyme is a catalytic RNA, R-I-B-O-Z-Y-M-E. That's a catalytic RNA. That catalytic RNA is a component of a structure we call a ribosome, R-I-B-O-S-O-M-E. Okay? That ribosome is bigger than a ribozyme. The ribosome has proteins, it has other RNAs in it, and it has this ribozyme that catalyzes the formation of the peptide bonds. Everybody with me? even if it's Friday afternoon? Questions about that? Yes? Does that ribozyme have a name? Yes, it does, actually. That ribozyme is called the 23S rRNA. You don't need to know that now. We'll talk about it later. But since you asked, I'm answering the question. Okay? Okay. Now, the smaller ribonucleotides I'm not going to talk about. RNA interference I'm not going to talk about, at least not right now, because, as I said, this is a little bit more involved. But these small RNAs that I told you earlier do help cells to control whether or not a protein is made. They play very important roles in that process. Okay, enough about nucleic acids in general. We want to turn our attention now to the synthesis of DNA. You've already studied some of this before, so it means I can go as fast as I possibly can move. Is that right? No, of course not. All right. If I say central dogma, how many people feel, I'm not going to ask you to do it, but how many people feel you could tell me what central dogma is if I said central dogma? 1? Okay. 2, 3, okay. I'll tell you what central dogma is. You hear this expression, central dogma. What the hell is the central dogma? Is there a central catma? Bad joke, yes. Yeah. Painful joke. No, there is not a central catma, I will tell you that. Okay? All right. The central dogma was something that they came up with when they first understood the relationship between DNA and protein. Now, I've briefly described that relationship to you already. I told you that messenger RNA was copied from DNA. And I told you that messenger RNA had the information necessary to make protein. There are single words that we use to describe each of the things I just told you. The synthesis of RNA is called transcription. One word, very simple. The synthesis of protein from that RNA is called translation. Transcription makes RNA from DNA. Translation makes protein from RNA. That's what the central dogma is. The central dogma says DNA makes RNA, RNA makes protein. That's the central dogma. It's not complicated. It used to be thought that that central dogma was a one-way street. You started with DNA, you ended up with protein. But by the late 70s, it was apparent that there were exceptions to that. And there's one of those exceptions is shown right here in purple. It's called reverse transcription. Reverse transcription means that in some cases, RNA can be converted back into DNA. In some cases, RNA can be converted back into DNA. Now, we'll talk about that again in just a little bit. But suffice it to say that reverse transcription occurs in some places that are, shall we say, less than desirable. There are, for example, viruses that have as their genetic material RNA instead of DNA. And in order for them to go through their life cycle, they have to convert that RNA back into a DNA. That is reverse transcription. It takes a special enzyme, and I'll talk about that later. But one of the viruses that has this property is a class of viruses known as retroviruses. Retro. We all know what retro is, right? Retroviruses go backwards. They go from RNA back to DNA. The most common retrovirus that you know of, you've heard zillions of times in your life, is known as HIV. HIV is a retrovirus. Okay. DNA replication. DNA replication means if I start with 2 strands, I end up with 4 strands. And so something must convert those 2 strands into 4 strands. That's schematically what you see happening on the screen. In the images on the left, what you see is replication of a circular DNA, like, for example, a bacterial DNA. The figures on the right show the replication of a linear DNA, like what would happen inside of your chromosomes. Now, it turns out that the considerations for replicating, that is copying, duplicating, whatever you want to say, the process for replicating a circular DNA is different than it is a linear DNA. Now, the overall process that we'll see is very similar. That is the copying of the strands. But how we process that is very, very different than what bacteria do when they have a circular guy. Okay? It's very, very different. Now, I'm just telling you that now because I'm going to remind you of that when I talk about the replication of eukaryotic DNA. And that's what you're seeing on the right, is eukaryotic chromosomes. Okay. Well, how does this overall process work? Well, first of all, like everything else that happens inside of a cell, reactions are catalyzed by enzymes. The enzyme that's important for the copying of DNA is known as a DNA polymerase, P-O-L-Y-M-E-R-A-S-E. You see it on the screen. A DNA polymerase catalyzes the reaction that you see starting to occur. It finds a base that is already paired to a DNA strand. So here's a base that's already paired to a DNA strand. It grabs another nucleotide, and it asks the question, is this nucleotide that I've grabbed complementary to the next base? If it is, it says, good, we're good to go. If it's not, it says, get out of here, and it grabs another one. Okay? So when it finally gets the right one in here, where the bases are base paired properly, it says, okay, we're ready to go. And what the DNA polymerase does then is it catalyzes the breaking of this bond right here between phosphates 1 and 2. And the joining of that phosphate to this hydroxyl group on carbon number 3 above. So we're forming a covalent bond between the oxygen, between this phosphate and this oxygen right here. That's what we're doing. That's what the DNA polymerase is catalyzing. It's making that bond I called the other day, that's the backbone of a DNA. The backbone of a DNA is a phosphodiester bond. Phosphodiester bonds, as I said the other day, link nucleotides. Peptide bonds link amino acids. So the enzyme that I just described to you, I'll repeat what it's doing. It's making sure it has the right base for the base pairing. It's breaking those 2 phosphates off that are on the triphosphate. And it's joining the phosphate to that hydroxide on carbon number 3. It's making a phosphodiester bond. After it's done that, the polymerase now says, okay, slide down one more. And it repeats the process over and over and over and over. Okay? You might think that's a pretty involved process, and it is a pretty involved process. That enzyme has to do a lot of different things there very quickly. Right? enzyme that I just described to you, that DNA polymerase in E. coli, can do what I just described to you: reading, cutting, joining, sliding down. It can do that process at the rate of 1,000 nucleotides a second in E. coli. 1,000 nucleotides a second. That's absolutely astonishing. And not only does it do it at the rate of 1,000 nucleotides a second, okay? It does it with greater accuracy than the most fantastic typist on the face of the earth. It will do that and make about 1 error in every 10 million bases that it puts in. That's pretty hard to fathom, folks. Now, we're going to talk about the process and how it manages to be so accurate, but it's pretty mind-boggling to think that that's what's happening and how rapidly that's going on. All right. Let's get to the meat of the matter. Okay? The meat of the matter. The meat of the matter happens at a place that we call a replication fork. A replication fork. Just like a fork in the road, we have a fork in the DNA. And if we have a fork in the DNA, that means something has to have pulled the strands apart. We're going to see that in DNA replication, there are several proteins that have roles. You've seen one of them so far, and that was the DNA polymerase. Some proteins, there's a helicase, for example, whose role is to pull the strands apart. It has to go fast enough to stay ahead of that DNA polymerase. That's pretty cool. Okay? We will see that there are proteins that help the DNA polymerase to stay on the DNA. The DNA polymerases will tend to fall off of the DNA. Okay? We will see there are proteins that help the DNA polymerase to get started making a strand. Because here's something that you'll find kind of shocking. There are no DNA polymerases that we have in our cells that can start the synthesis of DNA by themselves. None. Okay? All right. So that's what we're getting to here. Let us look at here. Now this is a much more complicated figure. What I'm going to do today is I'm going to spend about a few minutes describing what these protein functions are, and then we'll do a song for the day. Okay? And I should finish early. So I'm going to run this through you fairly quickly, and then I'll review it on Monday. Is that a deal? Okay. It turns out that at the replication fork, the first thing that has to happen is that the strands have to be pulled apart by something. This whole process that you see on the screen starts at a specific sequence in DNA. The proteins that do the replication recognize this sequence and start the process that I'm getting ready to describe to you. This sequence has a special name, and it has a special sequence also. It's called the origin of replication. All right? The origin of replication is where the strands first get pulled apart. The strands are pulled apart by an enzyme called a helicase, Helicase. The job of the helicase is to keep pulling strands apart ahead of the DNA polymerase so the DNA polymerase doesn't get slowed down. I told you that the DNA polymerase was moving at the rate of 1,000 nucleotides a second. That corresponds to 100 turns of the DNA per second. And if we multiply 100 terms per second times 60 seconds per minute, it means that these strands have to be pulled apart at the rate of 6,000 RPM. 6,000 RPM. The helicase is spinning and pulling strands apart at 6,000 RPM. That's faster than the engine in your car. People talk about molecular motors. This is an incredible molecular motor. It takes ATP energy to do that. All right? And if we think if we start pulling strands apart, what do you suppose is going to happen ahead of the place where you're pulling them apart if you have something holding the end down? It's going to get tangled as hell. If you don't have something to relieve that tension that the helicase is putting into the system, the whole thing will tie up in a knot. And it turns out that we have enzymes ahead of there that relieve that tension. The one in E. coli is called DNA gyrase, G-Y-R-A-S-E. And DNA gyrase is one example of a class of enzymes that we'll talk about later called topoisomerases. T-O-P-O-I-S-O-M-E-R-A-S-E-S. Topoisomerases. Okay? So the DNA gyrase here is a topoisomerase. So topoisomerase is a class of enzymes. But they have the property of, in this case, relieving that tension in the DNA. Everybody with me? Okay. Now, I'm briefly going to go over the other things that are here. I told you that DNA polymerase could not start a strand by itself. That is actually started by an enzyme called a primase, P-R-I-M-A-S-E. It turns out that at a replication fork in E. coli, which is what you see here, there are two DNA polymerases. One's called DNA polymerase 3, and another one called DNA polymerase 1. I'll talk about those next time. There's also an enzyme there called a DNA ligase. What does DNA ligase do? It joins together DNA fragments, and we'll see next time why that's important. The last thing that we have there that's of importance to us is this thing called single-stranded binding protein, or you'll probably call it SSB. Okay. Now, that's a lot of stuff. We're going to talk about the functions of those proteins in the next lecture. I promised I would finish early for you. And I thought we would finish with a song. It's called the Anthem for BB350. You can help me with this one by singing very loudly the 350 part. Oh, the students taking BB350 have an awful lot of things that we must know. With acetic acid buffer, Kevin Ahern makes us suffer. The exams could not be tougher. 350. There's amino acid side chains to recall. And the thing it takes to make cholesterol. Catabolic, anabolic, Kevin Ahern's diabolic. I'm becoming alcoholic. 350. There must be a way to jam inside my head all the metabolic enzyme names I dread. Can you help me learn the spaces where the endonucleases cut the DNA in places? 350. I must find a way to make a better grade, or my GAD will truly get waylaid. 350. I shall overcome frustration to achieve my aspiration on the last examination. 350. Here's the plan I made to help me to succeed. Fill the note card with the knowledge I will need. I put all of Ahern's quotes along with what each one denotes onto a massive stack of notes for 350. There is just one teensy problem I must fix. It requires some very skillful penman tricks. Squeezing info I must store onto the card he gave before will mean a font the size of 0.14. Oh, the student's taking BB350. Have an awful lot of things that we must know. With acetic acid buffer, Kevin Ahern makes us suffer. The exam cannot be tougher. 350. Okay. The good news on here is about the note card. On the final exam, I let you bring in a note card, but you have to write it in as small a print as you possibly can. By hand.